Platonic bacteria, Taurus spira, which is our model bacteria. It isolates from the onshore oil reservoir. But in nature, bacteria often live in biofilms, which consist a microbial community embedded in EPS matrix. The biofilm matrix can protect the embedded cells against environmental threats like flow-induced shear stress, temperature changes, or the chemical toxins like bicide. So the biofilm grows in flow systems can plug the flow path, create a plugging effect, which will cause serious problems in medical and many industry applications. So as the biofilm matrix can behave as a viscous fluid to resist the shear forces, to prevent the cells in the biofilm detached from the solid surface. And also the biocide treatment is very limited to its efficiency because the effect compounds are difficult to reach into reach inside of the biofilm to kill the cells embedded in the biofilm. So the biofilm tolerance to biocide is much higher compared to the Platonic bacteria. So because of the high resistance of the biofilm, so it's important to control the biofilm growth and detachment under flowing or dynamic conditions. So the traditional experiments like culture biofilm in a plate or in a flask tube, it's difficult to study the biofilm in dynamic conditions. So here we use the new developed microfluidic technology, which refers to precisely control the fluid on the reactant in a microliter or milliliter scale. So the microfluidics has become a very important platform for the biofilm research due to its advantage of real-time observation, and it's able to simulate the dynamic environment. So we can institute observe how the biofilm development in flow systems, and also the low reactant consumption. So here is our experiment setup. So we use this T-shape microchannel to simulate the biofilm growth in the flow systems. For this vertical channel, we use for the bacterial injection, and for this horizontal channel, we use for the nutrient injection. So during the experiment, we first inoculate the bacteria into the system through this channel for 24 hours, and then we close the system for 20 hours to let the bacteria have time to attach on the solid surface. And after 24 hours, we start to inject the nutrient solution to let the biofilm grow. So as our model organism, the Taurus spira, they can grow at ambient conditions. So the whole experiment was performed at room temperature and pressure. And the bacteria and the nutrient will meet at this cross section. So we place the microscope at this position and record the images of the whole biofilm growth. So in this method, we observe the entire life circle of the biofilms from the first initial attachment. We saw there are some biomasses attached on the glass surface, and during the nutrient coming into the system, the attached biomasses start to grow and form a biofilm. And the biofilm grows and getting mature, and then because of the shear stress, it might detach from the matrix. And as we are continually inject the nutrient, so the biomasses, they are going to another life circle. They regrow, and form the biofilms. So in our studies, we're going to study the injection and the surface conditions on the biofilm growth and the detachment. So like we study the flow velocity and the nutrient concentrations, how to influence the biofilm growth. And also we study the surface conditions like wettability and also initial attachment, how the initial attachment influenced effect further biofilm development. So these studies, 2 papers has been published, and also there are another 2 waiting to be published. But for all the experiment, we need a lot of data and repeat experiments to confirm our conclusion. But then we find that there is a problem. If we want to repeat the experiment in the same microchip, we need to clean the mature biofilms from the last experiment and clean the microchip for the next experiment. So it's a little bit difficult. So during our experiment, we find that the adhesive strength between the biofilm and the surface became stronger over time. So here is our experiment from the initial attachment to experiment. From initial attachment, and after 6 days, the biofilm grew to a larger size, and then we increase the injection rate step by step until around 6 times than the initial injection rate. But as we saw from the images, there is barely no detachment happened. So here we plot their biofilm coverage, which is analysis by the pixel of the biofilms. And we find that in these 2 experiments, when we increase the shear rate, there is no large detachment happened. And in experiment 1, in the beginning there's some increase, probably because of the increased shear stress promote the mass transfer between the biofilm, between the external solution and the biofilm inside. So it's difficult to find a way to clean the microchip for repeated experiment because we cannot use very high flow velocity. There is a pressure limitation for the glass chip, so it might damage the microsystems. And also we cannot use the strong disinfectants like acid solution. It will react with our glass chip and change the surface condition, which is one important parameter we're going to study. So the initial impacts for this study is we try to find the easy way to clean the microchip and without using the high flow velocity and harsh chemicals. So the previous biofilm researcher described a biofilm model. In the model, the biofilm is a viscous fluid, and the local high concentration of the the moleculars like cells, proteins, the byproducts, and the biomasses and the EPS, they create osmotic pressure differential between the biofilms and external environment. So the transport of water, gas, and nutrient into the biofilm is dependent on this osmotic pressure. So according to this, so the osmotic pressure could manipulate the transport of water and the water dissolved compounds across the matrix. So we can create osmotic pressure between the biofilm and the external fluid. So according to their theory, so we designed this experiment. We first grow the biofilm in the microchannel for 7 days to form a mature biofilms. Like here we show biofilm A, B, C. And then we start flow. We will create osmotic pressure in these 3 biofilms. So here we use the different salinity of the sodium chloride solution, which is very easy to make in the lab. So after the growth experiment, we first flood the seawater because the biofilm was growing in a seawater medium plus the nutrient. So just to use this flooding, we try to remove the residual nutrient in the microsystems. So after flooding this for 2 days, we start to inject the high salinity of the salt solution, 5 molar per liter. And as shown in the images, we can see all these 3 biofilms, they are get denser and compacted, and the first surface of the biofilms became wrinkled. So this is probably because the biofilm is dehydrated. And then after 2 days, we start to inject the 1 molar salt solution, and we can see as from images the biofilm A expand to a larger size, and also the biofilm C. But biofilm B, they not expand to a large size because there are a lot of the detachment happened. The biofilm C, they already detached from the original place and resettled in the new place. So from the image we can see the biofilm actually deformation according to the change of the external salinity. So here we plot the biofilm pixel, which is analyzed and the area of the biofilms during the injections. From the biofilm A, we can see it reduced by 34% after the 5M flooding, but increased around 4 times at lower salinity flooding. But as our calculation, around 63% of the increase is because of the resettlement of the other biofilms, because the biofilm A is located at the downstream of the microchannel. So there in the upstream, there are also have some other biofilms formation like biofilm C. So during the injection, they might reset, detached from the original place and resettled in another place. So there is 63% of this increase is because of the reset of the other biofilms. And the biofilm C, biofilm B is decreased around 17% at 5 molar, but reduced at 10% from 5 molar to 1 molar. This is because the variation of the salinity caused biofilm detachment. We'll explain this later. And the biofilm C is detached from the original place and increased by 136 during the 1 molar flooding. So the change of the salinity can force the biofilm to shrink and swell, and expansion in response to the osmotic pressure differential. To explain this is because, so in the beginning, the mature biofilm is create a osmotic balance with the seawater injection, which is the osmotic condition equal to 1. So the P in equal to P out. The P in the pressure in the biofilms, and the P out is equal to the pressure in the external environment. When we start flow the high salinity salt solution, 5 molar, which the osmotic concentration equal to 10. So the P in is lower than P out, so this causes the water exflux. And when they create another balance, we start to flow the 1 molar injection, which the osmotic concentration equal to 2. So here the P in is higher than the P out, so it causes the water influx. So from here we can see, due to the osmotic pressure differential delta P between the biofilms and external environment, the biofilm was observed to shrink and swell as the efflux and influx of the water cross the biofilm. To confirm our result, we performed another experiment. So in this experiment, we also first flood the seawater, and then we flooded the 1 molar salt solution. The osmotic concentration of the seawater equal to 1, and the osmotic concentration of the 1 molar salt equal to 2. So there is not much of the differential between the seawater and the salt 1 molar salt solution. And from the images we can see there is barely no change with biofilm E, but there is a lot of detachment happened in the biofilm D. So partly of this is mainly because in the beginning the biofilm D almost fully clogged in the microchannel. So the shear stress in around the biofilm D is much higher than biofilm E. So that might cause this detachment. And then we increase the salinity to 5 molar. You can see the both biofilms compacted and shrink to a smaller size. Then we decrease the salinity to 1 molar, and there are a lot of detachment happened in biofilm D, and the biofilm E expand to a larger size. So the biofilm E actually confirmed our result from the first experiment, which is expansion at low expansion at lower salinity, and shrinkage at higher salinity. And the variation of the salinity caused the biofilm detachment. Why? It might because these 3 reasons. First is because the water influx. The water influx leads to the reduction of the biofilm density and permeability and their adhesion strength with the glass surface, and also the connection between the biofilms and the glass surfaces might due to the ionic interaction and the van der Waals forces. So the changes of ionic strength of the external solution might change their interaction forces. And also another change is because of the surface charge density of the glass the glass is changes. with the local chemical environment. So these three reasons might cause the biofilm detachment happened. So here, from our cases, we try to find a new way to clean the glass microchannel. So from our experiment, like here, this is the mature biofilms. After the salt treatment, we left some biomasses, and then we flow the ethanol to clean the microchannel. Is only a few, only really tiny biomasses left in the microchannel, and which is very easily by increasing the injection rate to be removed from the microsystems. So for us, we find a new method to clean the glass microchannels for reuse the chip for repeat experiment, and without using high uh, injection rate and harsh chemicals to damage the microchip. And another aspect we want to test is we want to test to improve the efficiency of the buy-side treatment. As we discussed before, the buy-side efficiency in the biofilm is very low. But if we could create automatic pressure to let the biofilm shrink and the expansion to force the water plus biocide can influx into the biofilm and kill the cells inside. So, but unfortunately, we didn't test this in our study because the biocide might change our surface conditions of glass. So, if we have opportunity, we want to test this in the further studies. Also, there are other things in avoid to use the high amount of the sodium chloride. Like as we use a 5 molar sodium chloride, it might cause corrosion or the precipitation is somewhere. So we can try other high osmoactive compounds and also other microbial species. So even the physical principle of the how the osmotic pressure to drive the biofilm expansion and detachment is universal, but still the reaction time and the necessary osmotic pressure to initiate there to start to expansion and detachment are different. It's different with the species and the biofilm age, biofilm metric component. Like we reported the organism is Taurus biha, which is nitrate reduced bacteria. We also test another bacteria, Clostridium, is sulfate reduced bacteria in our system. So, but their reaction time and the osmotic pressure is much lower than Taurus biha. So, in the future, we need to test more bacterial species. So another aspect we want to study is we can use this method to manipulate biofilm to plug or unplug the flow paths in the flooding reservoir systems, like for the gas reservoirs for CO2 storage. During the gas injection, the biofilm can grow and plug in the injection path, which causes the injectivity reduction. But if we could inject the high osmotic, high salinity solution to make the biofilm shrinkage, so the permeability will increase. And after the injection, we want to close the reservoir to store the gas, so we have to block the gas leakage path. So in this we can close. So with this, we can inject the lower salinity solution into the tube to block their leakage path. So in the end, I want to thank our group member, and also this work, and also the conference committee and organizers. This work was supported by the Research Council of Norway and our industry partner. Thank you.